Okay, this is the second part of our digestive system lecture, and we are going to start by reviewing just for a moment where we were. We were just finishing up talking about the physical side of digestion, and we described how the mouth and the mouth was grinding up the food, how the stomach was churning the food and adding lots of moisture, but a couple of minor dissolving attempts in these situations, but not a lot of chemical digestion at this point. And what we just talked about was how bile is utilized by the digestive system in order to physically break down fat into tiny little droplets so that it becomes suspended and mixed with the water so that that fat doesn't stay in large clumps. So you can see from this diagram that there's the liver and the bile production, the storage in the gallbladder and the release. And then there's the pancreas as well. And what I want us to do right now is begin to talk a little bit more about now the chemical side of things, because this duodenum is where not only is this bile production going to be utilized, but where we're now going to begin to add lots of enzymes. These are going to be involved in the chemical digestion or dissolving of the food molecules so that they can be absorbed. So let's look at the pancreas in a little more detail. Now, the first thing I should tell you about the pancreas is probably that it's a dual gland. And by that we mean it's both an endocrine gland and an exocrine gland. Now, we haven't spoken as to what these two are, but let me just explain briefly. You might be able to tell from the endo-exo what you would expect. And when I ask this of students, most say yes. Endo is in and exo is out, like inside and maybe exiting. And that's exactly what it is. Endocrine glands and exocrine glands both produce some sort of chemical components. Endocrine glands release theirs into the human body, and that would mean they're being released into the bloodstream so that they can be distributed. Exocrine glands are going to release their substances out of the body. And it turns out that the pancreas is both. Let's look at a diagram that shows the cellular structure of the pancreas. If you followed those little branches in the pancreas out, this is where you would wind up. And notice that there is a blue tissue right here in the picture, and that there are blood vessels going to it. This is a little island of endocrine tissue, and these are scattered throughout the pancreas. These little endocrine islets produce hormones, and the hormone that is so important that's produced by the pancreas here in these little places is insulin. Most people today have heard of insulin and its relationship to diabetes. Well, it's the endocrine portions of the pancreas scattered throughout that produce that part. The pink parts that you see right here, like this, and this one right here, you can see that those are actually little pouches. You can maybe imagine that those cells are producing fluids, and that those fluids then are being collected by all the tubes. And just like branches on the tree, more and more tubes collect until you finally have that great big pancreatic duct running right down the middle of the pancreas. And these pink portions are the parts that are responsible for producing the digestive enzymes that the pancreas produces. And these are collected, as you see, and sent out of the body, aren't they? Where does that tube connect to? It connects to the duodenum. And of course, the inside of the duodenum is, that's right, outside the body. The inside of the tube is a surface of the human body, even though it's inside us. So these fluids are literally leaving the human body and entering the digestive tube. So that's the makeup of the pancreas. We will talk about endocrine glands later on in this class, but exocrine glands are going to come up over and over again. The salivary glands are exocrine. So that's the basic makeup of the pancreas. Now, just to speak of enzymes for a moment, since we've used the word a number of times, enzymes are produced not only by the pancreas, but there was one in the stomach, and there are many more here in the small intestine as well. The mucosa and the submucosa are both responsible for producing some of the enzymes that are going to help to chemically digest our food. Now, let's talk for a minute about what an enzyme is. When I talk to students about this, they often don't remember what catalysts or enzymes are. Without enzymes or catalysts, digestion could take forever. The enzymes that we're talking about are organic catalysts. I don't know if you remember what a catalyst is, but it's some sort of a substance or chemical compound that facilitates a chemical reaction, that speeds it up. A very simple description of a catalyst is kind of like what you see here in this picture. Here is a reaction, and they show it proceeding one direction. It could actually be proceeding in either direction, depending upon how the products and the reactants are being produced. But notice in this simple one, the catalyst, or in this case maybe the enzyme, is the green colored part, and the molecule that's being reacted with is the yellow part. And notice that the enzyme facilitates the breaking apart of the yellow substrate into two molecules. If we were to wait around for this just to happen on its own, it could take days and weeks and months for that to happen. But the enzyme does that and facilitates that and speeds that up. Notice that the substrate and the enzyme exactly match one another. The catalyst is not used up in the reaction. The reaction is that the yellow compound here is broken into two parts. And so the enzyme here, the green part, is a catalyst, speeds up the reaction. It can be used over and over again. It's not used up. It's not a part of the reaction at all. It just simply facilitates it. Here's another picture that illustrates the same thing. Here's an enzyme with an active site, and the substrate then attaches to it, and the enzyme facilitates its breaking apart. Now, realize that reaction could go the other way. The enzyme could be capturing two separate products and binding them together into one. Just look at the picture backwards instead of forwards. And either one is possible. And it is usually just dictated by the amount of whichever one is there. So, enzymes are substances that are not used up, that can be used over and over and over again, and that speed up a reaction like the simplification of, or the dissolving of, molecules. Let's talk about food for just a moment too. We're going to be real brief with this. There are 6 substances that are considered food: proteins, carbohydrates, lipids. You remember this refers to fats and oil type molecules, minerals, vitamins, and water. These 6 substances are what we consider to be food, that a person could not survive without some amount of each one of these. When you look at the makeup of, say, a protein, I've drawn it there, or a carbohydrate, or even some sort of lipid, a fat or an oil, you can see that they're made up of subunits. The individual little building blocks that you see there are referred to as monomers. And when they're all chained together like you see there, then they're called polymers. And the food that we eat are typically these complex molecules that you see here, these polymers. The digesting process is then to take and break apart these individual monomers, which are then useful to the human body. So we can illustrate it kind of like this. Here's a protein, a carbohydrate, and a lipid. So these are our polymers. And the digestive process is going to break them into monomers. So let's digest this protein. We'll add a protein enzyme. The enzyme will come along and break all the bonds between the proteins. The individual monomers are amino acids. And so we haven't destroyed the protein, we've just disassembled it into elements that can be dissolved and then absorbed by the body. We can do a similar thing to the carbohydrates. And this, of course, breaks them into simple sugars. Those sugars can be dissolved, whereas the carbohydrate could not. And finally, with the lipids, a similar action. The enzymes break the bonds between the monomers, and we're left with fatty acid molecules and a glycerol molecule. And these, again, can be taken up by the membranes of the digestive system and utilized in our human bodies. So that's just a simple look at how enzymes play a role in dissolving or simplifying foods. Let's go back to our intestine again. The duodenum, remember, was neutralizing the acids from the stomach and adding all of these enzymes and the bile. The enzymes then begin to dissolve the food, and the next step after that, of course, will be to absorb all of that food. The absorbing process really relies on there being a lot of membrane here. And the small intestine itself has a huge surface area. Let's talk about that. If you look inside the duodenum here where it's cut open, you can see that there are ridges, or what we call circular folds, that are present. And because the lining is folded, as you see there, it makes the inside much, much longer than the tube appears. Here's a picture that shows you the inside of that through a scope, and you can see the ridges there. Those are the circular folds. And so there's a lot more lining than it would appear. The folds are again illustrated as you see in this picture. Now, those circular folds then enlarge the surface of the small intestine. But not only that, but when you look at the folds themselves, the epithelial membrane is folded up and over, but it's also folded into little fingers or finger-like projections that are called villi. If you look here in the picture, the plicae circulares are what today we call the circular folds. And you can see them rising up. But more important, look over the surface of each one of those little ridges that's formed. And you can see that the very surface of the membrane is formed into little finger-like projections. This is an artist drawing here. Here's the micrograph, or the what it would look like under the electron microscope. You can see there the submucosa that supports the circular fold, and all the little villi then of the mucous membrane sticking out from that. Here's what each one of the villi look like in an artist's drawing. And notice that there are blood vessels up inside. And then if we take one villi, as you see here, and then take one little set, I think you can see clearly that there are columnar epithelia there. If we take one of those villi and look at it closely, we now see something else called microvilli. In the picture, you can see the basement membrane, you can see the columnar cells with their nuclei, and if you look at the cell membrane there at the surface, the cell membrane has been folded into finger-like projections as well. But this is at the submicroscopic level. Here's an electron micrograph showing those little villi that you see there, the microvilli. So we have not just folds and not just villi, but the cells themselves making up the mucous membrane have little tiny microvilli on their surface. Each one of these three is increasing the surface area of the entire surface of the inside of the small intestine. If these three things were not there, if we just had a smooth tube about 18 feet long, if we just had the smooth tube with no folds, villi, or microvilli, the total surface area would be about 4 square feet. With each of these three added to the surface, we have about 2,200 square feet. You can see that this is a huge increase. It's enough surface area to cover the floor of sort of a medium to large size house these days. It's an incredible amount of surface area that's folded into the inside surface of the small intestine. So, we've talked about the duodenum now. Let's look at the ileum and the jejunum. The jejunum is where most of the enzymes are added. And this is where most chemical digestion takes place. And most of the absorption also occurs here. So if you can imagine this food material passing through, it's this one segment where most of the work is done to chemically digest and absorb. As the food enters the ileum is just kind of becoming less and less of what the jejunum is. There's less villi, less circular folds, less enzymes. One thing that there's more of as you proceed along the ileum is there's more and more immune system structures, white blood cells, patches of immune tissue. And this is, of course, sort of guarding this area from anything that might come back from the large intestine. Not that the ileum isn't doing much, but since most of the work was done in the jejunum, the ileum is just going to squeeze out the last few bits of food. And so now, as the food exits the ileum and enters the large intestine, what we typically would see is the same nutrient material that went in your mouth, but with the majority of the nutrients dissolved and extracted from it. And so this is a waste product, but it's more like a leftover waste product. It's not really a waste product that's part of a chemical reaction, like exhaust from your car, or the carbon dioxide that's released from cells as they produce energy. So the food is the remnants of the food is now flowing into the large intestine. And let's just go back over the structures here of the large intestine. You remember the ileum? From the small intestine there, it enters at the side of the large intestine. There's a circular muscle there, an ileocecal valve, that attempts to prevent anything from the large intestine backing up. Not that bacteria couldn't spread through that valve necessarily, but that's where the immune function of the ileum helps to keep that at bay. The blind pouch-like end of the large intestine is the cecum, and proceeding then from there toward the anus is the ascending colon, and the food is through peristaltic action of the large intestine is pushed up. The transverse colon, it's pushed across. The distal sections of the colon, from the transverse to a flexure that leads down into the descending colon, that leads to the S-shaped part of the colon, the sigmoid. The straight part near the end is the rectum, and the last eliminating opening is the anal canal. And so there you have it. That takes us right to the end. And we should talk for a moment about the colon or large intestine. What is its purpose? Well, that's best illustrated with a diagram that you see here. The purpose of the large intestine is to reclaim as much water as possible from what's left in this remnant food material. Let's look at the statements here. So we're trying to absorb the remaining water from this remnant food material. Almost 7 liters of fluid has been lost in the digestive process. And by lost, I mean it was sent outside the body. And you know that's inside the tube. If you look at the picture, you can see that the salivary glands, and this would be in a single day, that the salivary gland, or glands, in a single day produce between 1 liter and 1.5 liters of fluid. Now, if you remember the liter, that is the same thing as a quart, or if you're more familiar with a 2-liter bottle of soft drink, half of one of those bottles in fluid flow from your salivary glands into your mouth each day. In your stomach, you can see there's about 2 liters of fluid. The pancreas and the liver together contribute about another 2. That 1.2 plus 0.7 is almost 2 liters there. And the secretions in the small intestine are another 2 liters. So 2, 2, 2, and 1 is 7 liters of fluid leaving your human body. Now, thankfully, it hasn't left to the surface. It's still in the tube, so we do have a chance to reclaim it. And if you look down below there at the red arrows, 92% of that fluid, of the water itself, is reabsorbed in the small intestine, and the majority of that in the jejunum. So the fecal material, the waste material, gets to the large intestine with still about 8% of the water that was originally added to it. That's still too much water to lose from your human body. You'd have to be drinking a whole lot more than you are today to keep enough water in your system. So the large intestine is going to extract the majority of that. And you can see at the end of the tube there, your feces should only have about 1% of the water that was originally added to it. So that is the major job of the large intestine, is to reclaim that water. The problems in this are twofold, and I've listed them both here: constipation and diarrhea. One is that the large intestine has taken too much water from this material. So by the time the feces gets to the anal canal, it is so dry that the friction between it and the canal is great, and it's very, very difficult for the material to slip out of the body. The opposite of that is diarrhea, where you have too much water and the fecal material comes out very, very, very wet. And when that's happening, you're losing a lot more water than you should from your human body. And usually doctors recommend, if you have an illness or something and you have diarrhea, that you should be drinking a lot of extra water to make up for that you're losing. And you can see here, illness can stimulate extra peristalsis. And really what's happening here is that the speed of the fecal material through the large intestine is what determines how wet or how dry it's going to be. If the material moves through the large intestine very quickly, right, it's not going to have time to absorb all the water from it, and you have diarrhea. If it moves too slowly through the large intestine and too much water is taken from the food, then that's where you have constipation. And you usually see that in people. If people aren't going to the bathroom at regular intervals, if there's long periods of time between that, then that's where constipation often occurs. And it's typically due to a lack of fiber in the diet. The large intestine and its peristaltic action sort of depends on having something to squeeze against. If the large intestine is squeezing and there's nothing there but fluid or something, then the large intestine is not stimulated to squeeze more. If there's a bulk, if there's an indigestible amount of fiber in it and it squeezes, and the thing it's squeezing on sort of pushes back, that stimulates the large intestine to squeeze more, and that's going to keep the food moving through the large intestine properly. That stimulation of peristalsis can sometimes be done with foods. Very spicy foods can stimulate peristalsis. And some foods do not. Foods that digest very easily. Plant materials are much harder to digest and tend to contain lots of fiber, whereas things like meats, when they're dissolved, there's very little material left after the dissolving process. Most of what's there is digested and used by the body, and very little left over. Something like a hamburger, especially if it doesn't have any veggies on it, like tomato or lettuce or whatever, can be very counterproductive because the meat is almost thoroughly digested. If it's a cheeseburger, the cheese is almost thoroughly digested. And if it's made out of a white flour bun that's had most of the fiber extracted from the flour before it was baked, then there's not much left there either. So foods, the kind of foods that you eat, if they thoroughly digest and there's very little roughage or fiber to the food, that's where we really, really have trouble. So we can explain a number of diseases and problems in what comes out of our rear ends by the amount of time that it takes for the material to pass through the large intestine. And so there you have it, a pretty good overview of the digestive system. In terms of processes, the digestive system is grinding and liquefying the foods. It's then dissolving the nutrients and then absorbing those nutrients, letting whatever's left over pass out of the body. Structurally, digestive system is basically a tube-like passage that runs from mouth to anus through the body. And accompanying that are a number of accessory organs. We've named these the salivary glands, the liver, and the pancreas. and they add fluids to the digestive process. So there you have it, the digestive system.